All right, my name is Clement. Welcome to this lecture on glycolysis. So today I'm going to be walking you through some of the whole steps of the process from glucose all the way to pyruvate, the products of glycolysis. So why is glycolysis needed? It is a breakdown of glucose that happens throughout the body that provides ATP to the cell, the main currency, if you will, to any cell in the body, honestly. Um, so let's talk about all of the reactions that happen in glucose, and I'm also going to mention some of the regulatory points that you should know of, and also one of the most common diseases that can happen in glycolysis and what the result of that is. So let's go through some of the reactions itself. So the first thing you'd want to know is glucose. Obviously, you probably know this molecule already. It's a 6-carbon sugar that's pretty much you take in through dietary means, or pretty much it can be generated in other ways as well. Um, so glucose, the first step of glucose and glycolysis is that glucose is moving around the cell. Okay, so glucose can come into the cell and leave the cell as it so pleases through the use of Na-independent transporters and Na codependent monosaccharide transporters. Okay, so these enzymes allows glucose to move into the cell and outside the cell as so it pleases, and this makes it hard to keep glycolysis going or carry out glycolysis because you need something to hold on to, something to restrict uh, further products of glycolysis from escaping the cell. Okay, so how does this cell do this process? So this happens within the cytosol of the cell, the regular fluid, not within any membrane-bound organelle or anything like that. So you take glucose in whatever concentration, and you pretty much add a phosphate to it using ATP and an enzyme called hexokinase. So hexokinase is going to be an enzyme that takes the phosphate group from ATP and adds it to glucose to make glucose 6 phosphate. So glucose 6 phosphate is now trapped within the cell because it has a charge that can't leave through this, the phospholipid membrane because it's very non-polar, the membrane is. Um, so this pretty much traps the molecule within the cell, and it also energetically favors it for glycolysis. By adding that phosphate, your energy toward that molecule is greatly higher. This step right here is pretty much a very exergonic process, and it can only happen in this one particular way. This next step is a little different. Okay, so this next step can actually go in both ways. But again, equilibrium-wise, you're carrying out glycolysis in one way as well. So let's just talk about it in a forward step. So once you have glucose 6-phosphate, you use phosphoglucose isomerase. This is the enzyme that's present, and it's pretty much going to rearrange the 6-carbon sugar, that's a hexose molecule, into a pentose molecule. So that rearrangement takes glucose 6-phosphate and turns it into fructose 6 phosphate. So once you have fructose 6-phosphate, you can use phosphofructokinase, another enzyme that takes the phosphate of ATP, puts it on this molecule to create a fructose 1,6-bisphosphate. Now this pentose molecule has a phosphate on two sides of it, creating this almost symmetrical molecule that you can break in half. So that's the next step that's happening in glycolysis. You can think of these 3 steps, these first, second, and third step of glycolysis as it's preparing the cell to the molecule to break in half, okay? By making it more energetically favorable. So this enzyme, hexokinase, is very exothermic, and this one as well is very exothermic. So so these two reactions do not actually go backwards. Fructose 1,6-bisphosphate is a molecule with 2 phosphates, and almost a symmetrical line that it can be broken. And this breakage is done by an aldose. This aldose is a carbon cleavage that happens within this molecule that results in glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These molecules have another name for easy um, saying. It's GAP and DHAP, as you can probably tell from how the full name of the molecule is. So what's important about this molecule is that these molecules, only GAP can actually go forward onto glycolysis. DHAP is a molecule that can't actually do that. So what the step that needs to be done is that DHAP has to turn into GAP, and this is done by a triose phosphate isomerase, an enzyme that actually doesn't have a very high negative delta G, so it can actually do the reverse reaction as well. This equilibrium favors GAP because as soon as you make it into GAP, GAP turns and goes forward with glycolysis. So thus, the DHAP becomes GAP with the use of triose phosphate isomerase. So once you have this GAP molecule, you can go ahead with glycolysis. So the next step in glycolysis is a step where you're generating NADH. So NADH is a molecule that's used further in the TCA cycle or the electron transport chain um, to generate ATP. It's an electron carrier that's used in the first complex of the electron transport chain to generate a proton gradient and transfer electrons across that electron transport chain. But you don't have to worry about that just yet because this lecture has to do with glycolysis. All right, so once you have GAP, you use glyceraldehyde 3-phosphate dehydrogenase. As you can tell with the last word, dehydrogenase, pretty much designates that it's going to be used to um, oxidize or take away electrons from the molecule. Okay, so it takes away electrons from glyceraldehyde 3-phosphate and puts it onto NAD plus. This is a very important reactant within this glycolysis, and makes it into NADH, this electron transport carrier. So once you do that, your result is 1,3-bisphosphoglycerate. Okay, now this molecule, another important thing about this reaction is that you have phosphate and it's going to get transferred onto the glyceraldehyde 3-phosphate. This reaction actually couples the transfer of electrons from NAD to NADH with the phosphorylation of this molecule into 1,3-bisphosphoglycerate. So what's important about 1,3-bisphosphoglycerate is that it can actually get turned into 2,3-bisphosphoglycerate, which you might have heard of as allosteric regulator of hemoglobin. And in red blood cells, this actually happens with the use of a certain mutase that turns 1,3-bisphosphoglycerate to 2,3-bisphosphoglycerate, and then it goes back into 3-phosphoglycerate. So the result of that is going to pretty much be the same, where you're creating ATP in this step. So phosphoglycerate kinase takes 1,3-phosphoglycerate, takes one phosphate from this molecule and puts it onto ADP, which is the reactant in this reaction. And that creates 3-phosphoglycerate. This 3-phosphoglycerate is not really useful in glycolysis, and so it needs to be transferred to or converted to 2-phosphoglycerate. So this molecule has its phosphate group on the end, whereas this one has it in the middle. So it's pretty much using a phosphoglycerate mutase, an enzyme that's going to take the phosphate from the end of the molecule and puts it onto the middle OH of this molecule, and creating 2-phosphoglycerate. So the second to last step of this reaction is the dehydration step with enolase that creates a double bond that favors the release of the phosphate group. in the final reaction, as I will tell you. So this dehydration by enolase will create PEP, or phosphoenolpyruvate. Enol, as you can tell, is a word that means like a double bond somewhere within the structure. So once you have PEP, or phosphoenolpyruvate, you can use pyruvate kinase, the final enzyme. It's actually a very important enzyme that makes more ATP, importantly, and also turns phosphoenolpyruvate to pyruvate. Okay, so we just went through all of the steps in glycolysis, but let me tell you which ones that are irreversible. So the reactions that are irreversible are the first step with hexokinase, the third step with phosphofructokinase, and the last one, everything else is actually reversible and done by an equilibrium constant as well. The only step that's not reversible, other than the first and third, is this last step with pyruvate kinase. As you can tell, you're creating ATP, and that has a large negative delta G. All of these irreversible steps has a large exothermic energy that's given off that results in a one-way favorable reaction. So what happens to this pyruvate in glycolysis? Why is it actually made? So pyruvate in cells with a mitochondria can actually go forward and turn into acetyl-CoA using a certain enzyme or complex called the pyruvate dehydrogenase complex. And this coenzyme A can enter the TCA cycle or the Krebs cycle and further be used to generate more NADH and FADH and GTP, which can make more ATP, and these electron transporters, such as electron carriers, such as NADH and FADH2, can go into the ETC cycle, or the electron transport chain cycle, to make more ATP, as I'll cover in the future lectures. So once you have pyruvate, so that's the normal step when cells that have a mitochondria. What happens to cells that don't have a mitochondria or are favoring anaerobic respiration, such as tumor cells, by at least a Warburg principle? So in cells that are favoring glycolysis or don't have mitochondria, pyruvate needs to be made into a thing called lactate. So lactate is made from lactate dehydrogenase. Lactate dehydrogenase. And you can kind of tell from the name, this is similar to this dehydrogenase in that it does almost a reverse reaction. It takes the NADH that's produced and makes it into NAD+. Why do we need to do that? Well, NAD+ can actually go back and be the reactant for this step right here, which is a pretty big step that's needed for glycolysis and the creation of pyruvate. So that's why we're making lactate as well. So what are some of the main steps that need to be regulated? Well, the irreversible steps. The main steps that are regulated, I would say, is this first step, hexokinase, the phosphofructokinase, and this final step right here, pyruvate kinase. And how do we regulate it? By looking at the products that are needed and the reactant. So if you have a reactant such as AMP or ADP, then it's most likely going to be favoring glycolysis. It's going to favor and be a potential activator for these enzymes. Whereas an inhibitor would be something like a product. Okay, so a product such as acetyl-CoA will be an inhibitor for pyruvate kinase. Okay, and this works by allosteric inhibition, whereas it binds to a certain difference site and alters the activity of the enzyme, resulting in a lower activity. And one important molecule to mention that's an allosteric inhibitor or activator in this case is actually fructose. We already have that molecule, so it's 2,6-bisphosphate. So fructose 2,6-phosphate, bisphosphate, is almost similar to fructose 1,6-phosphate, except instead of a phosphate in the one position on the first carbon, it's in the second carbon. And this is actually produced by a totally different enzyme, a phosphate kinase and a phosphate and regulated by a kinase and the phosphatase separately. That's also regulated by hormone levels of insulin and glucagon and epinephrine and such. So the main effect of fructose 2,6-bisphosphate is that it actually is an activator for this third step. So phosphofructokinase. You might think that for a fructose 2,6-bisphosphate might be an inhibitor because it's similar to the product, but it's actually a separate molecule. So this separate molecule is actually going to upregulate this enzyme, favoring this reaction to take place. And as I said, this is a rate limiting step. This is the biggest step in here that promotes glycolysis. So if you upregulate this enzyme, you're going to upregulate glycolysis. So as you can probably guess by this point, one of the main hormones that increases the concentration of fructose 2,6-bisphosphate would most likely be insulin. Okay, so that's one of the major things. Another major regulator for glycolysis would be, acetyl-CoA and other products, AMP, and then citrate can even be a very important, inhibitor for this enzyme as well. So, lastly, we went through all of these different reactions. And then let's look at the final one, the final thing that we have to look at, which is the most common enzyme deficiency that's seen in glycolysis. And that is, as you might think, is pyruvate kinase. Pyruvate kinase is actually the second most common enzyme deficiency, but the most common enzyme deficiency that's found in glycolysis. Okay, so this enzyme is a major one that you have to look out for. So if you don't have pyruvate kinase, what happens? You don't have an enzyme that's adding phosphate from phosphoenolpyruvate to ADP and creating ATP. Now, you don't have these two additional ATPs that's being produced from this step, creating a net ATP production within glycolysis as 0. Okay, so now you have 0 ATP production, and if you don't have pyruvate, so 0 ATP production, and no pyruvate, which cannot, regenerate NAD+, so that means glycolysis can't further happen as well. So 0 ATP, 0 NAD+, pretty much the whole glycolysis is useless because you're not making ATP, and if a cell doesn't have ATP, then you're out of luck. You're out of luck, okay? So that's gonna result in the cell dying. But this is generally not the case in every single cell. As I said before, this NADH can actually go ahead. NADH can actually go ahead and go into the electron transport chain. But what's important about the electron transport chain is it's only present in the mitochondria. And what are the cells that don't have mitochondria? Red blood cells. So red blood cells are the first to get activated by or affected by this deficiency, and that results in destruction or hemolysis, and eventually resulting in anemia, so hemolytic anemia. So thank you for paying attention to this lecture, and if you have any questions, please leave those in the comments, and I will get back to you. And thank you for visiting my channel. Please leave a like and subscribe for more educational videos, as well as future videos on anything else that might be needed, or even an introduction to myself, actually. So thank you.